got a couple of minutes there to get yourself familiar with the Tropilaelaps mite that you can see there harassing a Varroa mite. And this is quite a broad talk covering a huge body of work, so I've kind of got to squeeze it into a bit of a nutshell, really. And I'll say my thank yous at the end, but just to mention our sort of mainstay throughout, this has been a sort of two-phase project, but our mainstay here has been bee disease insurance. So I just thought I'd mention and sort of thank them preemptively at the start for their support, because they've supported us right from the beginning, actually, and without them this project would have never even got off the ground. So just an early thank you to BDI. So what we're going to talk about here, we'll just talk about what Tropilaelaps is, a bit of the morphometry, some of the distribution, where it is and what's going on, some of the effects on the hive, and what the National Bee Unit does, the surveillance it does, and some of the current detection methods and research that we've been doing. And I'll also give you an overview of a survivability study that we've been working on. And some work that we've done this year that is very new, so it'll be a brief overview of that as well, and some of the future research. But I'll just mention this picture here because it's kind of might be intriguing to some of you, but that is actually a Varroa mite and a Tropilaelaps on a pinhead, which took some getting, I can tell you. So it was my attempt at being sort of somewhat artistic and trying to sort of capture the precipice of the struggle between Tropilaelaps and Varroa mite, and I wanted to maybe capture them on a tightrope or something, I don't know. But anyway, I went with a pinhead. It took a long time to do, and I think I kind of captured it in the end, because I wanted to demonstrate the sort of the slightly more powerful but more agile Tropilaelaps with the cowering Varroa mite down the side of it. So anyway, I just thought I'd throw that in there. It's kind of something that I wanted to add, and yeah. No mites were harmed in the making of that picture, by the way. And just to introduce you to the team, so like I said, this is a project that's happened in two phases over 23 and 24. So the team that have worked on both phases of this are within the picture there, but Victoria Tomkeys from Fairer Science has worked with us on phase 1. Maggie Gill, she's a senior science officer for DEFRA, and also this Fairer Science here, so she's been on both phases. George Tong from the National Bee Unit, Paul Davies from the National Bee Unit, and Avra Royal, who was involved in phase 2 National Bee Unit, retired National Bee Unit. So what is Tropilaelaps? Well, it belongs to a genus of parasitic mite called Laelapidae. And that parasite is from a very big family with 1,300 described species, and it's a parasite of vertebrates, invertebrates, and it's in free living forms as well. So its original host is the giant honeybee species, which is Apis dorsata and laboriosa, which is this absolutely stunning bee here, found in Southeast Asia and broader. I've got a map to show you that in a second. And they do these large single comb nesting activities normally on buildings or tree branches and so on. So there's 4 subspecies, only 2 of which are noted to be harmful to Apis mellifera. And that's Tropilaelaps mercedesi, which is widely spread throughout Asia, and Tropilaelaps clari, which is more confined to the Philippines, but is spreading. And obviously both are known to be a severe threat to Apis mellifera, and known to vector honeybee viruses. And I just thought I'd quote this here. This I don't know if you can see my pointer very well, but this is this Laelapidae. This paper published in 2024 described that Varroa are actually part of the Laelapidae family as well, because that was in dispute for some time. Just in case you're interested, I thought I'd add that in. As I said, it's successfully adapted to parasitise Apis mellifera, and now considered a serious threat, jumping from the species of the larger honeybees. So it feeds on the hemolymph of larvae and pupae. And again, I've quoted a paper there from Han, which was actually where they'd done some work with Varroa. So that was where they discovered that not only do Varroa feed on the fat body of adult bees, but actually they are feeding on hemolymph of pupae and larvae, and during that experiments that they were doing, they had found out that Tropilaelaps also does feed on the hemolymph of larvae and pupae. So it causes malformations and transmission of pathogens and population decline. Lives most of its life in brood cells. It can come and go freely in unsealed cells. Thought not to be able to feed on adult bees. We've got a bit of evidence to suggest that may not always be the case, but it is likely. And some evidence to show survival for longer periods on young nurse bees, certainly dead bees and decaying larvae. So we have had mites living for quite a while on those things. So Varroa spends 3 to 13 days, with an average of 7 days on adult bees, at its phoretic stage. And Tropilaelaps can spend 1 day, even less than 1 day, maybe hours. It invades the cells at the 3rd, 4th and 5th instar phases. Can leave the cell at any time. Again, that's obviously unsealed cells. So there's more genes related to pesticide resistance than Varroa, and possible alternative hosts. So I've quoted a paper here again. So this was from 1961, as you can see, and it's a very small paragraph within that paper that says that in an apiary that was dying out from mite infestations, they'd found mites on rats within that apiary. So that's kind of interesting, more work needed, but like I say, it just really mentions it as a kind of a side note in that paper. And this is just to compare the two, shows you the difference here between Varroa on the top, Tropilaelaps on the bottom, and it's almost twice the reproduction rate of Varroa mites. So I then imaged the ticking time bomb. So that the concern with this mite is the speed in which it can expand within the hive. So some of its characteristics: it is 8-legged. So you see these two extended prolegs at the front. They're not antennae, they're actually prolegs. They're very hairy, lots of sensory receptors on those legs, seemingly probably almost double what are on the other pairs of legs. It's an elongated body, 1.5 millimeters in length, 0.5 millimeters in width, and it's this light reddish brown. And you can see there the difference on the Varroa mite here. So it's about 1/3 the size, and it doesn't reflect the light in the same way that Varroa does. You've all sort of noticed on your pull-out floors, you can even if your eyesight's not that good, you can kind of make out the sheen that Varroa mites have. They're kind of this slightly reflective. Tropilaelaps don't do that. They're a very sort of dull brown. They're much harder to see, and that will be the overriding theme of this presentation, is really just how small and hard to see they are. So this is a couple more images here, just looking at the difference between male and female for one. So you can see on the right here, this ventral plate and anal shield, the slightly two different shapes. It's not always that evidence, but this blunt end here versus this pointy end is the slight difference that we can tell males from females. Gravid to non-gravid females, so that's egg-carrying females to non, so they have this expandable body, kind of like, I always think of it as one of those sort of like hard-shelled suitcases that you undo the zip around the middle and it pops open. That's so they don't expand out, they sort of expand up. And this here is a good evidence of that, and we've got other images of that too. So eggs are about 50-50 male-female, which is kind of interesting, unlike Varroa. So obviously they can, increase genetic diversity very, very quickly. Damage to the hive, kind of similar to Varroa mites, really. So, the comb that you can see here, so it's perforations and chewed down pupa and larvae like you would see for Varroa mites. And they tear holes in the larvae as well. They're quite damaging. Unlike Varroa, very polite feeders kind of using the same feeding sites. And Tropilaelaps don't do that, and I think it's the next slide that you'll see that is evidence of that. But they will, by doing so, they're scarring the larvae, damaging the larvae as they're nibbling on it all over the place. So I've said this, it reproduces much faster than Varroa. We've already talked about it, vectoring honeybee viruses. And colonies are known to collapse at a faster rate, and that we've had more and more evidence of that just in recent times. Again, like I say, may be able to feed on young bees. That's still kind of out there for some debate at the moment. And this is a paper produced on the feed site, so they've used this dye here on the left-hand side there, so that's they've been able to look at the feeding sites of Tropilaelaps, so you can really see there that they're really making a mess of that larvae. And on the right here, this is how it manifests into these malformations and even, you know, damage to the tergite. This is antenna there under electron microscope, you know, holes in the antenna, so it really can be quite damaging, and you know, you can just see the mass of marks that it's making over this larvae. So some of the viruses there. So this is a study from South Korea, and you can see that it's largely the same as Varroa, but this is quite interesting here that deformed wing virus was in 100% of the Tropilaelaps that were studied. But no black queen cell virus, which was quite interesting. Just a little video here, you can see a bit of movement. You can see those pro legs at the front, you know, these hairy little legs that it's using for making its way around the hive. And they do tend to do this, they surf around the cell. And again, I can see that that's a pregnant female, a gravid female that's body's distended with eggs. And just a bit of it here on the comb, so you can see a bit of movement, how fast it is. You saw on that first slide how fast the mite is compared to the Varroa. It's kind of like looking at a whippet run round an aged St. Bernard or something. You know, it just moves. It's so much faster than a Varroa mite. And again, just surfing those outer perimeter of the cells. So just a bit of distribution here. So and a couple of questions, but it's not even in Europe yet. Well, we're going to change that now because you know, we know that it is making its way over, and it is actually in continental Europe. And it won't be able to survive British winters. Well, we'll talk about that in a second as well. So what's interesting here is this blue on the map is the native range of Apis dorsata and Laboriosa. It's their home range. That's the native range of Tropilaelaps. But the red is the spread of where Tropilaelaps is now found. There's some areas there unconfirmed. Data out of North Korea is a bit thin on the ground, so we're you know, it's unconfirmed in that location. But kind of very interesting that the amount of spread outside of its native range. And Pakistan actually reported huge losses of Apis mellifera as well when it made its way to Pakistan. So this is just got the updated map here of Europe, so you can see that it is over into several Russian provinces. We'll look closer at that in the next slide. Where the stars are is obviously where we are now finding Tropilaelaps. So this is provinces right up against Ukraine. Obviously, in this area, there's bigger fish to fry than surveillance for Tropilaelaps at the moment, but we are right on the border. This is Dagestan on the Caspian Sea. That's the original discovery of Tropilaelaps within Russia. So as of the day before yesterday, which we've been expecting for some time, it's now been confirmed officially in Georgia as well. Now, Iran is down here. last year it was suspected it was in Iran, then the report came out that it wasn't, so that's somewhat a bit unclear about exactly what's happening there as well. And I just thought I'd quote this paper from Georgia because it is that new. And also, I'll read you a quote here, actually, off of that paper, because it just captured me. But it says, our findings revealed high infestation rates of Tropilaelaps mercedesi, co-infestation with Varroa destructor, and notable mite reproductive success. These results underscore the threat posed by Tropilaelaps mercedesi to Georgian apiculture and highlight the potential for further spread across Europe. Immediate action and vigilant monitoring by national and international authorities are crucial to mitigate the impact of beekeeping and apiculture. So I thought that was quite a poignant quote to bring today, and like I say, that is fresh off of publication. So this is a paper from South Korea. We just talked about British winters. Well, in Seoul, they can have temperatures down to -10 quite frequently. 96% of the colonies contained mites across the region that they tested, 85% Varroa, slightly less with 76 of Tropilaelaps, but 68% of all the colonies did have both mites present. And again, so we've got average temperatures here of Seoul in South Korea, so we're right down into the negative numbers here, and we put Glasgow on there just to see, just to compare to. So in terms of temperature, obviously, you know, that doesn't seem to be a factor, and we'll just talk about that on the next slide, which is this paper that was produced in 2024, and this was some work that was done in those Russian provinces. And again, this is the temperature range here, well below zero for winter temps. And it was these two provinces up close to Ukraine here. What's really interesting about this is that the orange here shows the infestation of mites, and the blue is the brood that was present in the hive. So you're looking at around about 200 cells of brood present during that winter period, and 0 mites. So the work that they did at this time showed that there was no mites on the bees, no mites in the brood, even though then in April, so we started off with a couple of mites, and then May, and that's what's amazing to me, is that growth rate is huge. So you've gone from no mites to lots of mites, and the question is really, where were those mites? And that's really some of the work that we've been trying to pursue. What were those mites doing? Where were they? How did they go from 0 to 60 in no time at all? So some of the routes into the UK, obviously transport and shipping is a big one. We've had Apis florea colony on a shipping container one time. Obviously queens are probably a serious factor for us. So, and this was also some Apis florea that arrived in Malta just recently. So that was, you may well have seen that. So things are certainly on the move. This was an interception into the UK here. Now I was intrigued by this because I didn't actually know what it was until I realised that this is an Apis florea colony that we've taken in Thailand, and actually that lump there is the lump of honey that is in this box. You can actually see the twigs through the middle of them as well. So that was intercepted in the UK. This is honey, packaged honey from various. I think this was sent to me from a colleague. I'm not exactly sure where this was, but this is obviously imported honey. I'm not actually sure that is Apis mellifera comb in there, to be honest, but you know, things like this are making its way into the country. So what does the National Bee Unit do about it? Well, we've got an exotic risk program. So we will be doing inspections around these risk points. So these are all scored risk points around the UK where we'll be doing honeybee inspections. And we've also got a sentinel apiary program. We have two. We've got a voluntary type, which is beekeepers volunteer to take part in, and they'll send us samples. And we've also got an enhanced sentinel apiary that the bee inspectors will tend to and monitor themselves in conjunction with the beekeeper, of course. And then we do nearest neighbour inspections. So we'll be inspecting all the apiaries around as many of these high risk points as we can. So the detection methods as it stood, I should say stood, we would normally examine the brood cells and the comb, floor scrapings. We're going to talk about this more as we go on. The comb bump method, we'll look at that in a second. That's off some of the really good work that Dr. Jeff Pettis done some years back now on how to detect for Tropilaelaps, and visual inspection of the floor inserts as well. And we'll talk a bit more about that as we go through. This instigated our research trip to Thailand that BDI and others graciously funded, and really sort of saw an opportunity to progress with this. So in 2023, a group of us went to Thailand, and we did some work here in Chiang Mai and down here just south of Bangkok, and with the team here. And as I've quoted there, we know enough to know we don't know enough, and that's really the situation we are in. So the impetus behind that trip was really to test our current standard operating procedures. Do they really work? Can we detect Tropilaelaps within the hive? And if Varroa can be kept alive, now this is just me personally. Many years back actually, I was keeping Varroa alive in a jar in my bedroom, strangely. So I know young men keep strange things in their bedrooms. I was well over 14 days, and I don't remember the exact time frames. It was a long time ago, but Varroa just seemed to be able to survive, and I just couldn't believe it at the time. So again, when we're thinking about Tropilaelaps, we really need to look at what their survivability was like. So we wanted to look at the potential routes into the UK. We wanted to look at how the mites survive, the mites survive to make it into the UK. So a survivability study was really on the cards there. And how likely is the lab to pick it up if the samples are going into the lab as well? Is that going to work? So we wanted to look at the efficacy of the bump method, and really seeing the pest in the field is the only real way that our inspectors, all we're going to learn about this mite, and our inspectors are going to be able to see. So Thailand is a great place to study Apis species. It's got a great range of them, including Apis cerana, is the Asian honeybee that most of you would have heard of related to Varroa. Apis florea is these lovely little, so cute little dwarf honeybees, and of course Apis dorsata, the ones that we think that Tropilaelaps originated from anyway, and the black dwarf honeybee as well. And obviously the European honeybee is an import into Thailand, just in case you were interested. I'm sure you knew that. But actually Thailand closed its borders to imports of honeybees some years ago. So they've got a very nice strain of Ligustican, nice Italian bees there that seem to be holding true to their breed very well. We then, so the study took place at Chiang Mai University. Obviously, we looked at 65 colonies across 6 apiaries. So really that was, can we detect mites? That was really the main aim. And obviously, Thailand is different from the UK. It's got a different way of keeping bees, but it's basically the same. But some of these intriguing hives that they've got here, which is things like this storage box, which was actually made quite a good hive that held a Langstroth frame. Oftentimes they are this is a palm oil plantation here. This here is hives that will in rings around fruit trees and so on. So we're working in quite nice conditions often. And also the bees are that nice that you can work them in short sleeves and a t-shirt as it goes. And we'll talk more about this in a second actually, but bear that in mind, that little stick with a cotton wool bud on the end of it. So obviously our main aim at that point was could we just find a mite? So we were working this is the university apiary, and fortunately for us, there is Apis dorsata just hanging on the buildings 4 to 5 floors up, just right next to the apiary. So that was quite convenient too. We're actually able to be in the classroom and watch and collect samples from Apis dorsata, which was very convenient. But But eventually, after some looking and some shocks, when we just realised how hard these mites were to see, we found 0001 Terry the Trop, which was, we were all celebrated to find one, and it was quite a good moment for us. But it really was quite worrying that we that we sort of struggled to find them when we thought they might be everywhere, you know. So that is the bump method. It's a clean white tray. Tap the frame on it. Do mites drop off of it? It's as simple as that, really. And obviously, this is you know, you do get the odd mite drop off of it. It does work well to get the odd mite to come off. We're going to talk more about that in a second, but that is essentially the method as it stands. So obviously, these hives are slightly different. Already said, as the floor is fixed to the bottom of the hive. So in order to get a mite count, we had to design these floors very quickly, as you can see, a scribble on a piece of paper. And each of these hives then had to have all the frames removed, this floor laid into the bottom of it, and all the frames put back in just to do the monitoring and so every time we wanted to take the floor out, it was a whole process to do so. But we obviously very quickly realised that, that inspecting a floor in any condition like this or close to this to find a tiny speck of dust that doesn't reflect the light in any way, like I just said about Varroa, was all but impossible. And then to be able to scrape this material off and put it through the Endicott sieves within the lab and still try and find this mite within the small specks of detritus was almost impossible. So we kind of deemed that not really a viable method to find the mite or to be able to surveil for the mite within the UK. And the bump method, we realised that it did also cause a fair bit of damage to the brood. So this is where one frame would have been replaced back into the hive. And then within 24 hours we've pulled that out, and that's the brood that the bees have been picking out over that time period. Uncapping, so we were uncapping drone brood and uncapping general brood. That would be another method that we were previously using. Firstly, the mites don't seem to show that much of a preference for drone brood. You know, that's not scientific, that's just an observation. And the other thing as well, with the Tropilaelaps being so soft and small, the amount of sort of exudate junk and juice you get from a uncapping drone brood or any brood is would hinder you being able to see the mites. So we settled with this 10 by 10 rhombus for so we would individually pull out the larvae and pupae. And we also wanted to trial these new methods. So this is the Varroa Easy Checks, and this is, we wanted to use a familiar method to most beekeepers, 300 bees in an alcohol and a sugar and CO2, and I'm sure most of you know what these units are. So obviously we set to work on this across all the 6 apiaries we had. We had a very efficient system where everybody stuck to their same job so that we could record the data very clearly. Everyone was doing the same thing at all turns. And this was how we were first doing the sugar shakes. So we'd shake the sugar out over onto a white tray and tease through it with an artist brush to try and find the mites. It worked, but we've moved on slightly from there, and this is inspecting the bottom with the magnifying glass, trying to distinguish between the bits and these tiny mites. So the results of that were the fact that we ended up with uncapping and sugar. As you can see here, the uncapping was very successful, and actually sugar turned out on top. I was quite surprised. I thought alcohol was going to be really good on this. It still maintained relatively well on the Varroa, but not for Tropilaelaps, interestingly. CO2 wasn't really working well for us with Varroa or the Tropilaelaps. And the bump method we did release some mites with, but again the damage to the frame was of concern to us, that if we were rolling this out to beekeepers in the UK, they might not be quite so keen on. So we've actually ended up going with this 2-prong approach here. So you'll see your bee inspectors, if you are involved in our Sentinel Apiary programme, you'll see the bee inspectors doing a combo of these two methods to try and find Tropilaelaps or surveil for Tropilaelaps in the UK. And with the icing sugar here, that is collected and goes to the lab for analysis. So actually this here is a photo of our very first sample of 2024 that came in from one of you guys, I'm sure, in this room. And that's all your Varroa there that we can see in there. So we're able to get a really clean sample from the field, which is great for us because that can go straight to the lab and we know that it's contained within a bag and it goes through this system and is able to be cleaned efficiently, and we know for sure now that we will pick up Tropilaelaps if it is within that sample. And this is the paper we produced off of that study, so you're welcome to go and peruse that. That's just been recently published, and we've got 3 more papers actually on the way. So current treatments, just as an overview, this is a Chinese fluvalinate treatment. So this is a paper strip that they lay in front of the entrance or just behind the entrance as the bees walk in, and they'll walk over this and get fluvalinate on them and pass it around the hives. And this is for real, taken from Thailand. This is a dog flea treatment mixed up in this jar here with this little fogger. The translation was a bit vague. We're not exactly sure if they were spraying them on the bees, on the frames, down the seams, or exactly what was going on. But this is a home mix of flumethrin. And then this here is a cotton bud on the end of a stick that is soaked in formic acid, and that's put in the hive as well. And just quoting a couple of papers here. So this is Dr. Sam Ramsey's paper here on the use of formic acid in 2 forms, Formic Pro and a generic liquid, and also some heat pads. So they had quite a bit of success with treating Tropilaelaps with formic acid. I think the heat pads showed a fairly quick recovery rate. And then on the right here is a different paper. This shows the use of Baverall and queen removal and queen caging for Tropilaelaps. And just the highlights are the Baverall seemed to work, seemed okay and reduced the numbers of Varroa, but it was not nearly as good on the Tropilaelaps with the Baverall actually. Interestingly, the brood breaks did seem to have an effect though. So again, that's a gravid female, an egg-carrying female, and just observationally, we've noted that they are the mites that are the most mobile. They're everywhere. They run around and they're running rings around those Varroa, kind of harassing them a bit. But it does tend to be those egg-carrying females that are doing that, which is quite interesting. There we go, the Varroa sort of ambles off, all disappointed. The in vitro study, so essentially this is what we've nicknamed it the pot study, but this is essentially looking at mites on stuff, if you like. So we'll put different matrices in pots and look at the survivability and longevity of the mites. How long do the mites survive? Could they survive long enough to reach the UK? So just to set this up then, so really we've got several different scenarios that we worked on here. So mites on brood, mites on dead bees, mites on fabric and clothing, newly emerged bees we're interested in, and for queen cage attendants and fondant, so we sort of mocked up a queen cage, mites on comb and mites alone, of course. And obviously to do that we needed mites, and we needed lots of them, and as I said at the start we struggled very slightly, and that was until we met Mr. Bum, who's the beekeeper here in the middle, is his real name, and he did fortunately have 1 or 2 mites for us, unfortunately for him but fortunately for us, but he certainly provided us with what we needed, which was great for us to start the study. So obviously this involved offering Mr. Bum some money for his frames, and we took them off back to the lab and we set about harvesting the mites from the frames. So we were aiming for 20 mites per pot. 30 would have been better, but there was 3 replicates of each treatment group, so that was the aim. And you can see here, the volume of work that goes into that, busying away. So a couple of things that we realised through this is that, we learned a lot along the way, and harvesting mites was a lot more difficult than we thought. They move really fast. The pooter and the aspirator was probably the best way, so we're literally hoovering the mites up, and an artist brush we often use to move the mites around as well. But the one thing we noticed is if you got them wet or sticky, they struggle to recover. They're not very robust, unlike a varroa that seem to be able to plough through things a bit more. Tropilaelaps would be just sort of stuck and glued to surfaces really easily. And obviously we needed 420 mites for the first experiment, 360 for the second, so we collected a total of 780 mites. So it's a lot of work just involved in that, actually. And this is just a small demo of what we needed to do that. So it's a slightly modified aspirator or pooter with a drinking straw to kind of narrow the aperture slightly, and you can see the mite there that just disappears up. So that frame there, you can see it's all been uncapped one by one. The mites are released, and hopefully, if we're lucky, we get to suck them all up cleanly and not to suck up the honey and sticky stuff. You get any moisture in there, and it ruins the whole sample. So we normally empty that out when we've got maybe 20 or so mites in there, and then we move on. Like I say, it's a lot of work. So they're all divided up into their individual pots, and they go into the incubators, 33 degrees, 60% humidity, and they're counted. The first experiment we did this twice. So the first experiment we did, we counted at 24 hours, and we realised we lost a lot in that period. So we on the second one, we counted at 1, 3, 5, 21 and 24 hours to try and capture that the death rate of the mites. And obviously that included all days and nights and weekends, trying to do everything else as well. Counting mites on anything is not easy, let alone live bees that run around and all the rest of it. That's so not easy, but it was something that we just persevered with and carried on with. It needed a microscope most of the time to just be able to see, to get a good visuals, particularly on the bees. But obviously on larvae, they're obviously much more visible on the white background. So the lessons learned from pot study 1 was that bees move dead mites. We would have to mark the mites, and we could then record that the bees would easily pick them up and move them around. They got stuck in everything. They crawl up the sides of the pots and get stuck in the lid, so we had to make some amendments for that. They go in the feeder, and also we inverted the pot, so we've done something slightly different. We used a fluon liquid, a PTFE liquid, and that stopped the mites can't climb on that very well. Like I said, we reduced the count time as well. So pot study 2, then we reduced our matrices. So we basically decided on the stuff that we really thought we wanted to focus on. So we had slightly less replicates that time on the second time round. But so this time we had mites on brood, mites on dead bees that we'd frozen. That's how we killed the bees. And mites on newly emerged bees. And mites with the adult bees and the fondant to replicate the queen cage again, mites on comb and mites alone as the control. And again, it was three of 60, so 60 mites per treatment option, and counted at those time intervals. So just a very brief summary, but really mites die very quickly without the bees or larvae. As you can see here, mites alone tend to die very quickly in a group. But really, the take-home here is that on the larvae and the emerging bees, so they seem to die very quickly. The first 50% died very quickly, and then after that, they seem to last a lot longer. So it's kind of interesting on the emerging bees, So the question is, did they set up some kind of feeding site once in a limited area that maybe only half the mites could access or something like that? And also, we were asking ourselves, do males, females, gravid females, do they fare better on emerging bees? Stuff that we need to look at, questions we still need to ask. But as we've said there, that mites do die alone quite quickly. but they can survive on emerging bees. Oh, very dead and decaying bees and larvae as well seem to have a bit of longevity, and we'll look on that on this one as well. So again, I said this was a broad overview. I've thrown in the results from this year, so this is 2024, and we did the study again and actually added a lot more variables as well. But really interesting here is firstly the control, you can see, died very quickly. So this is the time period across the bottom here in hours. The control bees, as we would expect, died very quickly, but this time we took some Apis dorsata samples, and they died not far after was the Apis dorsata. The mites died on those very quickly. So we can only really maybe assume that the dorsata were killing the mites, because otherwise that's really, at this point, it's the only feasible thing that shows why they died so quickly. So were they nibbling at them and biting them within the sample pot? But obviously the longest here, so this is dead bees. So this is bees that were, rather than frozen this time, they were just squashed. And obviously when you squash a bee, you get lots of juice and you know, soft liquid out of the dead body. And likewise with the purple line here is the dead pupa. Likewise you get more moisture from that as well. So it certainly seems like decaying and dead stuff the mites are lasting much longer on. And so the dead adult bees was 192 hours, which is 8 days. So much longer than we would have thought, the survivability. And the other thing we did this year was a miticide resistance test. So they have shown resistance to the commonly used miticides, and also this oxalic treatment here. I wanted to do a small pilot study for oxalic sublimation this year, so we also did that, which actually showed in the pilot we did 87% reduction in mites in the brood, which was obviously a really interesting result for us, but needs a bit more work. And just a very quick overview of that oxalic. So you can see here the red is the untreated colonies, so you can just see a huge spike. And again, that's always been the concern with Tropilaelaps, is how quickly they exponentially grow. And the blue line is the floor from the drop on the floor. Again, so we had to do the same as last time, where we put this floor into the hive each time to check the drop. And we had to leave this, obviously we had to leave the mite drop so that the oxalic, what we didn't want to happen is we didn't want to capture the drop that we would expect to get from the treatment. We only wanted to capture the drop we would get from the effect of the treatment, if that makes sense, because you'd obviously expect to get a drop from the oxalic itself. And we just, this is the uncapping here, and we just got a little spike there, and I remember that very vividly on the day, that we just had one hive that actually had quite a lot of mites within the brood, and obviously that sent our data out with a big spike, which was, you know, it is what it is. But yeah, so overall 87% we thought was quite good on the efficacy of the oxalic sublimation. And then just the quick results of the miticide as well. So you can see amitraz was the most effective, but it's only really effective at 64%. So that's still very low. That's only just above what you would consider real resistance. And this was just a schematic of how we had the experiment set up. But fluvalinate, flumethrin, particularly the fluvalinate, and as you saw from the previous slide with the Chinese treatments that it's quite widely used in Thailand, but certainly showing some resistance there. And some of our future work, several articles, obviously keep a lookout for. They're being released slowly, but we are releasing articles in various magazines. 3 more peer-reviewed papers in process, so we've already published one. We've got a lot more to do from that. So we want to expand on the in vitro study, looking at why the Tropilaelaps are surviving so much on adult bees. Obviously, we want to look at why the Tropilaelaps are surviving in these temperate climates, what's actually happening. You know, remember the slide back in Russia there where the mites just grew very, very quickly. Are they entering some kind of diapause? Are the eggs able to enter some kind of diapause? You know, what's actually happening there? And more understanding of the mechanisms within the hive and, of course, on Apis dorsata. So we need to understand how this mite is working on its native host would be a good place to start as well, because there's such a lot we don't know about that yet. So the efficacy of the organic acids, so namely the oxalic, that needs more work. So the pilot study we did needs to be expanded into something slightly bigger and better. And trialling the efficacy of approved UK Varroa treatments as well is another thing. So lots going on for future, and obviously we'll continue to gain as much material as we can in terms of video and photography. So that's just from another feeding site observational experiment we were doing, but just more for the fun fact that we were marking mites there. And let me tell you, marking mites is quite a feat. And you can see that bead, they're trying to brush them off and get rid of them. They do tend to gather here on the thorax there, and certainly in between the abdomen and the thorax they sort of congregate, and we noticed they did that on the Apis dorsata as well. That's generally where they all went to collect together. So really, in conclusion, Tropilaelaps are more damaging, more prolific, harder to see than Varroa, and can overrun a colony relatively quickly because that growth curve grows far quicker than Varroa. They are coming, detected in Europe for the first time, and obviously that's now being released, and momentum amongst researchers has been gaining for some time on this, but really this is about bringing beekeepers on board a lot more. It's not about fear-mongering, it's about sort of education and awareness, really. The behaviour is not fully understood. Overwintering, we just talked about that. Treatment is difficult. They've shown resistance to synthetic chemicals. You know, we need to understand dosage rates a bit more. And of course, you can read our paper on the detection methods as well there. And just before you go, there's another one I want to mention, and that is Varroa. So obviously, Varroa is a different mite, part of the Laelapidae family as well. This is a parasite of Apis florea. There are two subspecies there. So we've got Varroa sinhai and Wongsiri. Obviously these have been studied over a period of time. There's a paper here from the 70s, but there's so much that's not known about Varroa. They have been able to survive on Apis mellifera larvae within the lab, but also anecdotal evidence that they are in Apis mellifera colonies as well. So we have heard of that. We haven't seen it yet, but this one here is quite interesting, is that it can live for 50 days on worker bees outside of the nest. Again, that's not work that we've done, but it is work from the past. So, you know, not to kind of put the wind up you, but there is another one out there, and there's just another thing to look forward to. And just to leave you with a Tropilaelaps mite doing a backflip. There you go. And then that just leaves me to say thank you very much, and thank you to all the supporters again, particularly BDI, who have been there right from the start. This would have never happened without them. Wax Chandlers have supported us. The National Bee Unit and APHA have supported us. You can find more out about obviously everything bee related at thenationalbeeunit.com. And for our future work, please visit firoscience.org as well. And this is Bajri Chutong from the Chiang Mai University in Thailand that's been a massive support as well. So I'd just like to thank her too.
With the oxalic treatments, did you do the sublimation just once, or did you do multiple?
Sorry, multiples, yeah. Sorry, I didn't mention that. So every 5 days for 20 days, but we did the count at 25 days.
It was interesting, the resistance mechanisms you've seen there coming through. And you said you were going to be testing the approved miticides for the UK market. There's a few categories there which aren't present, which because they have multiple mechanisms of actions, could be interesting to consider. And these include the essential oils, thymol and some of the other essential oils. Have you considered those as a future, or just
Yes.
for prioritisation?
Yes, no, exactly, yeah. Yeah, no, we would like to, all of the approved UK miticides would be worth doing more work on. Trouble is with thymol is that obviously it involves a lot of exterior environmental conditions as well, so heat mostly. But it seems like the work that Dr. Sam Ramsey and so on had done with formic, which is obviously also something that we'd be concerned about in high temperatures, seems that they did seem to have some success with that. So, yeah, we would be looking at everything, but that's something, future work that we really would like to do down the line. That's not something that we're doing right now, or of really. We've got nothing in the pipeline specific for thymols or that type of thing, but possibly for the future, sure.
And there's a further category, and that's the differential sensitivity of mites, particularly varroa mite, to heat, and the fact that a 2-hour temperature hit with 60% humidity causes a huge mite drop. There's work being done again in Germany and some more in the States on that. Yeah,
well, again, that Sam Ramsey's paper there had the heat pads on there, so it might be worth you looking at that paper, actually. I think you'd be interested to have a look through that paper.
Yeah. Okay, thank you.
Thank you.
I have one question for you, actually, Dan.
Mm-hmm.
Obviously, all these treatments are designed to go on the hive on the bees, but when we're treating varroa, obviously we don't have the supers on and honey and the rest of it. So if the build-up is going to be 2 to 3 months, we sort of the treatment frequency is something that troubles me.
That's a worrying thing for me too. And interestingly, I think with the oxalic acid in particular, that's something that could be, we've looked at that for multiple treatments. It does seem to have an effect. Obviously, that needs more work, but that might be something that more, because the growth rate is what it is, a more frequent type of treatment, but less stressful and less impactful. I don't, really that the whole system probably needs to be looked at if we're going to, start thinking about the frequency of treatments, and or a completely different mindset from varroa as well, possibly.
I noticed the stars were rather close to Turkey, and I think of Turkey as being rather a vibrant beekeeping country.
It is.
Have you had any contact? Whether they're jumping up and down, and if so?
Yeah. I'm in touch with some researchers in Turkey actually, and yes, I know that there is a migration pattern between, quite close to the borders in the east, and they migrate really far to the west and southwest for pine, I believe it is. So Turkey is a concern for sure. There's some very high mountain ranges, I believe, through, and I think it mentions it in the Georgia paper there. So they're kind of they're wondering if that could be a bit of a barrier for it moving south out of Georgia. But obviously we know these things. Eventually somebody moves some hives somewhere. But as far as I'm concerned, I do believe it will get from one end of Turkey to the other end of Turkey pretty quickly, if and when it gets there, yes.
I can remember beekeeping way before varroa, and varroa was really devastated beekeeping. It seems that Tropilaelaps is going to be far worse than that, and the possible, or the greatest possible what way it will come in will be with importation. So surely we should be discouraging beekeepers from importing bees.
That's probably not for me to answer that, Roger.
I'm not expecting you to. I'm not expecting you to, Dan. But for beekeepers in general,
I think it's a great point, Roger, for sure, because it probably, the thing that we've got that a lot of other people in Europe haven't is a bit of ocean, a bit of wetness between us and the mainland, and obviously to me that seems like a very sensible barrier, and obviously importation of queens is something that is obviously going to be heighten the risk. I think that's all I probably can say on the subject, actually.
Well, I did say discourage.
Thanks, Roger.
I was just going to ask how robust our testing is of imports coming into the country of honey and comb and bees. Do we test everything, or is it just...
No, what we do test is the queen cages. So all the workers are decanted at the point of entry, and the cages are all gathered together, and the packaging material, and that all goes to FERA to be tested. So yes, that does happen. Obviously, there's a time issue there. Maybe you know, what then happens to these queens? Is the queen importer recording movements in the best way they can as well, in case something does need to be recalled, you know, because you don't know if these things are going to, the materials will go to the lab, and then on the same day queens are being distributed across the country. That all would then have to be recalled, and you know, but yes, that's certainly for queens, honey and that type of thing. I'm not entirely sure that's certainly not within our remit to work on that, so I wouldn't like to comment on that, actually. But bees, yes.
To me, the weak point is not necessarily bee or queen imports. It's the fact they disappear from the hive for 2 or 3 months. If we don't know where they go, we don't know what to protect against. And that could be a far bigger problem, potentially.
Yeah. And that's an area that interests me or us as a team hugely, is what is going on there, and that's why I think we need to look at, so Apis dorsata are a migrating species, so they move around with the nectar flow, so they'll have broodless periods. You know, we know they don't survive well off of brood, but something is happening somewhere that they're able to survive either in the environment or wherever it may be, but something is happening there where they're able to survive for long enough that they can then repopulate quite quickly.
The question was going to be, is anyone looking into this?
Honestly, I don't really know the answer to that. That's some of our future work. You saw it on our future work slide. I don't know what some of the other folks are doing, and I would imagine that some of the researchers, there's some great researchers out there that is certainly becoming more on the radar now. And like I say, for researchers, it has been for some time. And like I say, Jeff Pettis, Sam Ramsey, those types of guys have been working on it for a while. I can't comment on what work they've got in the pipeline, but for us, it is something in the future we're hoping to look into, because it's you know, certainly up there in the top 3 things that we're most interested in.